Griffith proved that there is something called as a transforming principle which converts R-type bacteria into S-type. Now, he did not know what the transforming principle was. It was later on that Avery, MacLeod, McCarthy in their experiment and Hershey and Chase in their experiments proved that Griffith's transforming principle was the DNA. DNA is the transforming principle, was first proved by Avery, MacLeod, and McCarthy. So let us see what their experiment was. Avery, MacLeod, and McCarthy, 16 years later in 1944, Oswald Avery, Colin MacLeod, and Maclean McCarthy attempted a more definitive experiment. Now, what did they do? They took the two same bacteria, Diplococcus pneumoniae R type and Diplococcus pneumoniae S type. From the S type, they purified DNA, RNA, and proteins. So, from the S type bacteria, they removed DNA, they removed RNA, and they removed proteins. And from the heat killed S type bacteria, DNA, RNA, proteins were removed and they were mixed with the R type to see which could transform living R type into S type. And they found only those mixed with the DNA were transformed into the S type bacteria. So, from the heat killed S type, जब डीएनए निकाल के आर टाइप के साथ मिक्स किया गया, आर टाइप with the help of DNA formed a capsule around it and got transformed into S type. So, that transforming principle is not a part of RNA, not a part of protein, but only a part of DNA. And then what did they do? They took all the three materials. They took RNA, DNA, and proteins and put it into a test tube. RNA, DNA, protein, put it into a test tube. RNA, DNA, and protein, and put it into a test tube. And ये है RNA, DNA, and proteins, RNA, DNA, and proteins of the S type. And उसके अंदर they put live R type. Live R type was put. Now they put over it RNAs, RNA destroying enzyme. So RNAs would destroy the RNA. And then they put protease. And then they put DNAs. And when they put RNAs, they found RNA got destroyed, but still live R type got converted into S type. When protease was put, still, what will RNAs do? Destroy RNA. What will protease put? Destroy proteins. And what would DNAs do? Destroy DNA. Now, when RNAs destroyed RNA, R still got transformed into S type, which means when RNAs destroyed RNA, RNA में gene for capsule नहीं था. And though RNA was destroyed, R got converted into S type. When protease was put over this collection, protease must have destroyed the proteins. So DNA or RNA were constant. DNA, RNA were constant. DNA or RNA as it is rahe, and proteins were destroyed by protease. Protease destroyed proteins. So protease destroyed proteins. But Proteins were destroyed, but DNA, RNA was as it is. And R got converted into S type. R type got converted into S type. So when RNase was put, it destroyed RNA, but R got converted into S. When protease was put, it destroyed proteins, but R got converted into S. Now, when they have put live R type and they have put DNA, RNA, and proteins, now they put DNase, which destroys the DNA. DNA destroys ho gaya, and as DNA is destroyed by DNase, they found R did not transform into S type. R did not transform into S type. What is the meaning of it? When DNA is destroyed DNA, DNA के अंदर gene for capsule must have been destroyed, and as gene for capsule was destroyed, the R type did not get converted into S type, which conclusively proves that not RNA, not proteins, but DNA is the genetic material. ये अच्छा एक्सपेरिमेंट था, बट ये थोड़ा सा प्रिमिटिव एक्सपेरिमेंट था. A much more significant experiment was done by Hershey and Chase in 1952, proving that DNA is the genetic material and not proteins using bacteriophages. So Hershey and Chase proved that DNA is the genetic material and not proteins using bacteriophages. What was Hershey and Chase's experiment? Hershey and Chase worked with viruses that infect bacteria, that is called bacteriophages, which composed of DNA and proteins. And check this here. A bacteriophage attaches to the bacteria and its genetic material enters a bacterial cell. The bacterial cell treats the viral genetic material as its own and subsequently produces more virus particles. So, ये है एक bacteriophage, and this RF sir will teach you in diversity of life. When it attacks a bacteria, it passes its genetic material inside. And once its genetic material is passed inside, using its genetic material, more daughter bacteriophages are formed, and it will help in rupturing of the bacteria. Bacteria rupture ho jayega and daughter bacteriophages are liberated. So a virus will attack a bacteria. Virus will attack a bacteria, pass its genetic material inside, and once its genetic material is passed inside, the bacteria will use virus's genetic material and form daughter bacteriophages. The bacteria will rupture and daughter bacteriophages are liberated. So that is the way a bacteriophage, a virus, uses a bacteria as a host cell to produce daughter bacteriophages, which are then liberated. So this is something that Hershey and Chase knew. Now they wanted to discover whether the genetic material which was passing from bacteriophage into the bacteria was DNA or proteins. Now, what did they do? They used radioactive phosphorus P32 in a medium for some viruses and radioactive sulfur S35 for some other bacteriophages. तो P32 भी यूज किया है, S35 भी यूज किया है. What is the idea of using these two isotopes? Phosphorus P32 goes in the DNA. Phosphorus is a part of phosphoric acid. तो P32 यूज किया तो DNA becomes radioisotopic. And when proteins are used, Proteins are not a part of DNA. Proteins contain sulfur. सुना है proteins are connected by disulfide bonds. बायोमोलिक्यूल में पढ़ा है आपने, प्रोटीन्स आर कनेक्टेड बाय डाइसल्फाइड बॉन्ड्स. तो सल्फर S35 will become a part of the proteins, and some viruses were with radioactive DNA and some with radioactive protein coat. And ये है एक बैक्टीरियोफाज जिसके अंदर हमने उसका प्रोटीन कैप्सूल होता है. So what does a bacteriophage consist of? It consists of a capsule and a neck and tail fibers. And capsule is proteins. तो कैप्सूल प्रोटीन होता है, बीच में होता है डीएनए. तो जब आप S35 यूज करोगे, then the S35 will become a part of the capsule. And जब P32 यूज करोगे, it will become a part of the DNA inside, not the capsule. तो जहां S35 यूज किया है, the capsule has become radioactive, S35. और जहां P32 यूज किया है, the DNA becomes radioactive. The DNA becomes radioactive. Now, S35 में capsule has become radioactive. P32 में DNA has become radioactive. It was allowed to infect the bacteria. तो दोनों बैक्टीरिया को इन्फेक्ट करने के लिए डाल दिए. Now, what happened? It was found daughter bacteriophages were formed, and जहां S35 यूज किया था, the bacteria के अंदर there was no radioactivity detected. Bacteria में no radioactivity was detected. And जहां S32 यूज किया था, P32 यूज किया था, radioactivity was detected inside the bacteria. So, what must have happened when S35 wala capsule was allowed to coat? What really happens is a bacteria passes its DNA inside a bacteriophage passes the DNA inside the bacteria, and the capsid is out. It goes away. Capsid चला जाता है, and उसके अंदर S35 की radioactivity मिलती है, which is in the supernatant. So you put the bacteriophage over the bacteria, allow it to centrifuge. Bacteria being heavy will sediment, and the capsules being light will become supernatant. So, where did we find the radioactivity? In the sedimented bacteria, there was no radioactivity. जो सेडिमेंट हो गया और जो सुपरनेटेड हो गया, उन कैप्सूल्स के अंदर रेडियो एक्टिविटी डिटेक्ट की गई. सो वेयर वाज द रेडियो एक्टिविटी? इन द कैप्सूल्स. सो कैप्सूल्स में रेडियो एक्टिविटी हो गई, which means capsule has not become a part of the bacteria. And jahan bacteria with P32 was allowed to infect, and capsids went out, and in the bacteria centrifugation was done. Bacteria के अंदर radioactivity detect की गई, and the supernatant, the capsules did not have in it radioactivity. So who has actually gone inside? S35 अंदर गया या P32 अंदर गया? S35 तो supernatant में डिटेक्ट हुआ. So it does not enter the bacteria. तो अगर S35 supernatant में है, it has not infected the bacteria, means proteins, the capsid bacteria के अंदर गया ही नहीं है. And proteins cannot be the genetic material. But when P32 was allowed to enter the bacteria, and you got bacteria sediment के अंदर रेडियोएक्टिविटी है, बट कैप्सिड्स में रेडियोएक्टिविटी नहीं है. तो S35 in the initial experiment, S35 got radioactivity in the supernatant, but the bacteria did not have radioactivity. Bacteria did not have radioactivity because इसमें P32 नहीं था. And second experiment में bacteria had radioactivity because it contained P32, the genetic material, and the supernatant did not have radioactivity because S35 तो था ही नहीं वहां पर, and proteins became part of supernatant. S35 became a part of supernatant in the first experiment. When capsids were there with S35, the radioactivity was found in the supernatant, not inside the bacteria. And when P32 was present in the bacteria in the form of DNA, it is the bacteria which showed in it radioactivity. Bacteria में radioactivity मिली, supernatant में radioactivity नहीं मिली, which means that capsid containing S35 were part of supernatant. उसमें रेडियोएक्टिविटी मिली एंड बैक्टीरिया में रेडियोएक्टिविटी नहीं मिली. तो proteins cannot be genetic material. But when P32 DNA was radioactive, and when the bacteriophage attacked the bacteria, then it must have passed its DNA inside, and the capsids became supernatant. और DNA यानी बैक्टीरिया के अंदर रेडियोएक्टिविटी डिटेक्ट की गई, इट वाज रेडियोएक्टिव, which clearly proves that DNA containing P32 is a part of the genetic material. उसकी मदद से daughter bacteriophages बनेंगे, and this was a nice conclusive evidence to prove that proteins do not enter into the bacteria. They are supernatant, which means they cannot be effectively carrying genes. And DNA is having genes and is seen inside the bacteria as P32, means segments of DNA are called as genes. So, three experiments done, which scientists gave the transforming principle. Transforming principle was given by Griffith. Griffith gave the transforming principle. Which scientists proved that Griffith's transforming principle is the DNA. Griffith's transforming principle is DNA was proved by Avery, MacLeod, and McCarthy that Griffith's transforming principle was DNA. Who used radioisotopes and bacteriophages to prove that not proteins but DNA is the genetic material. Hershey and Chase used radioisotopes and bacteriophages to prove that not S35, in other words, not proteins, but P32, in other words, the DNA is the genetic material. Nice experiments, and we end it with the modern concept of genes. So, this was given by Seymour Benzer. Genes are units of inheritance. A gene is a segment of DNA that provides instruction for synthesis of a specific protein or a particular type of RNA. So, gene forms the RNA by transcription, which forms a protein by translation. A gene is defined as a segment of DNA responsible for inheritance and expression of a particular character. A gene is responsible for inheritance and expression of a particular character. Seymour Benzer introduced the term cistron, muton, and recon. Cistron is the unit of function. It is responsible for expression of a trait. So a gene undergoes transcription and translation to form a protein and expresses itself. Then what is that functional gene called as? That functional gene is called as a cistron. A cistron is a functional gene which is responsible for expression of a trait. It is a segment of DNA having information for synthesis of a particular protein or RNA. So it forms an RNA which forms a protein. It is several hundred base pair long. So what is a gene nowadays scientifically called? A gene is called a cistron. तो ये वर्ड हमने पहले से किया था. Humans have one gene forming one protein which are called monocystronic. And bacteria में multiple genes undergo transcription to form a single mRNA, and that forms multiple proteins with polyribosomes, and that is called polycystronic. So एक functional gene को एक cistron कहते हैं. Then we have muton, a unit of mutation. It is that segment of DNA which undergoes a mutation. तो डीएनए का छोटा सा सेगमेंट जो म्यूटेशन करता है is called as a muton. It is only few nucleotides. Could be one point mutation. जो एक म्यूटेशन है वो पॉइंट म्यूटेशन, और few base pair say 3 or multiples of 3 frame shift mutations, and these are the smallest units of expression, mutons. And then we have recon, unit of recombination. It is the segment of DNA that participates in recombination during crossing over during meiosis. So pachytene में that segment of DNA which crosses over and undergoes recombination is called as a recon, which is a few to many base pair. So एक जीन जो एक्सप्रेस करता है उसे क्या कहते हैं? सिस्ट्रॉन. एक जीन जो म्यूटेट करता है उसको क्या कहते हैं? म्यूटॉन. A gene which undergoes crossing over or recombination during meiosis, during pachytene, what is that called as? Recon. So cistron, muton, and recon are the modern concepts of gene suggested by Simon Benzer. Chaliye, with that, we have understood the experiments, the modern concept of gene, and with that, we have now finished off with the most important chapter of your entire syllabus, the toughest and the most decisive chapter, molecular basis of inheritance. इसे पढ़ना नीट में इसमें से सुपर क्वेश्चंस आते हैं. मजा आएगा आपको. चलो, टेक केयर, गॉड ब्लेस यू.